introduction, and thank you all for coming to hear what I have to say. We do collaborate for a long time, but I don't communicate with all of you. So some of you know most of the things we do. Many of you don't know much. So I will make a relatively extended introduction into glycans because they are relatively unknown to the majority of people. And then I will focus a bit on what we know about glycans as the modifiable biomarkers and functional effectors of the age-related diseases. So the way I like to look into glycans is that they are adding an additional layer of complexity between genes and proteins, because the majority of proteins are actually glycoproteins. So they will have a polypeptide part, which is defined by a gene. So it's an evolutionary wisdom for billions of years. And then there will be a glycan part, which is a part of a structure, which does not have a gene. It's encoded in a network of dozens or even hundreds of genes, which decide which kind of glycan is going to be attached to a specific glycosylation site. And adding a different glycan, alternative glycosylation, is functionally analogous to coding mutations, but it's inherited as a complex trait. So we can change the structure of a protein without changing a sequence of nucleotides, without changing a sequence of amino acids. And one nice example of this is the collaborative cross of mice we studied together with the Grant Morhan. So he started with the 8 founder strains, crossed them for 3 generations, and then inbred the second generation cousins into the strains. And then we looked at IgG glycome into these different hundreds of strains, and some of the strains completely separated from the rest of the mice. So they were structurally and functionally different without changing a gene sequence, just by reshuffling the allelic variants and having a complex traits which generates this diversity. In addition to this genetic diversity, glycans also integrate epigenetic information. So everything what we learned from our prenatal and postnatal lives, but also the environment, what we are eating, what we are thinking. And all this integrates into this complex network, which decides which glycan to put on a specific protein, which can then have different functions. And for example, if we talk about the glycan age, which I will talk about later, approximately 40% of it's heritable, while the rest is mostly lifestyle related. And glycans are actually the ultimate layer of molecular complexity. And I like to use this analogy here of these two birds. So the protein revolving in the middle is the S-glycoprotein of the SARS-CoV-2 virus, and everything hairy on its surface are the glycans. And it's very hard to understand function of a protein if we ignore these hairy things. So bird with the feathers, that's a glycoprotein. And this poor little bird who lost the feathers is a polypeptide part. And we could be the best scientists in the world and had all the funding and planned the experiment perfectly. But if we ignore glycans, if we study the featherless bird, the closest to the understanding of flight would be this bird walking. So we cannot understand the flight if we have a featherless bird, and we cannot understand the protein function if we have proteins without the glycans. And maybe one of the best examples is IgG, because all the effector functions, at least vast majority of the effector functions of IgG, so what will happen after IgG binds to its target, are determined by the glycans attached to this Fc domain. Different glycan, different structure, binds to different effectors, different receptors, and have a different effector function, either having pro-inflammatory, which is kind of an intuitive way of thinking what immunoglobulins do, but also being anti-inflammatory. So if we put wrong glycans, antibodies actually start to ignore the antigen and behave in an anti-inflammatory way. And the majority of proteins are glycosylated, and the majority of proteins are in some way regulated by glycosylation. And to understand this full spectrum of function, like this bird here, we have to include glycans. So one word of caution, because before I move further, glycation is something completely different. So glycation is a chemical reaction between glucose and proteins. We usually quantify it as HbA1c, which is then being converted to advanced glycation end products. This is the random damage. This is taking a shotgun and firing at proteins. Glycosylation is a sophisticated modification involving hundreds of genes, which creates these modification of proteins. And protein glycosylation was instrumental for the development of multicellular life. This is something what Lauc and I published as a hypothesis paper over a decade ago. And I think finally we managed to get some solid evidence. This is still unpublished, but when we did the phylostratification of all the genes involved in glycosylation, there was a big peak of glycoprotein evolution in the period when life was becoming multicellular. So when we were developing multicellular functions, then the glycoproteins evolved and took over these functions, which they also do now. So it's a fantastic evolutionary tool. But also, this is the way how we are being made different, how each of us is a little bit different, because we are all not the same. And if you look just in genes, we are nearly the same if you look in the gene sequence. But when we look into the glycans, and these are just 6 random individuals with the composition of the IgG glycan, different color, different glycan, clearly people are different. And this is affected by our genetic information, epigenetic information, and environmental factors, which integrate into diversity. And this diversity is functionally relevant. And maybe the best example are the antibodies. You have probably all heard about rituximab, one of the blockbuster drugs. It has one tiny little problem. It has the wrong glycosylation. It has glycans with this core fucose, which is a functional inhibitor of its function. So one of the most sold drugs on the market is being inhibited by approximately 95% using this core fucose, which people were not aware of as a problem when rituximab was developed. So recently, a variant of a drug called ublituximab was developed. The only difference is it does not have this core fucose. So it has glycans without a core fucose. And it's 100 to 500 times more potent in vitro assays, but also in vivo, just because it doesn't have a wrong glycan. And glycoengineering, designing the proper glycan for a drug, is becoming a huge business, and all big pharma is now investing into glycoengineering because having the wrong glycan can really prevent function of protein from having a normal function. And one of the key reasons why glycans are being ignored by the majority of researchers still is that this is very difficult to analyze, because each glycosylation site can have multiple different structures. So each peptide would separate into tens or dozens or even hundreds of glycopeptides. And there can be also multiple glycosylation sites on the same protein. So recently there was a paper describing a protein with over 1 million different glycophores. So extremely difficult to look into, especially in high throughput manner. And we were the first who went in that direction. We published our first high throughput glycomics paper over 15 years ago, and then followed this up with a large number of different grants, mostly European. We managed to get over 40 million euros, which we focused into the glycomics and developed into the largest glycomic group in the world, which generates approximately 80% of the global high throughput glycomics output. There was a recent review in a Chemical Reviews, which listed 191,000 published glycans in a large studies. Out of these 191,000, we generated 159,000 glycans, over 80%, which we generated in my lab. And we did it in collaboration with many leading researchers around the world. And actually, the largest glycomic data set is in the TwinsUK. And we use it a lot, but I think there is a potential to also do much more with the data. And I think one of the key information here is that we really generate the high quality glycomic data. This was an interesting study where we were following one famous scientist who collected his samples on a weekly basis for 10 years, and we analyzed all of them. And in 795 samples, there were only 4 outliers, which were 10% different from the past point. So it's a combined analytical and biological variation was below 10% for 99.5% of samples. So it's really high quality analytical data, which can be then connected with all other data and generate knowledge about how, why, and when are glycans being changed. And we published a lot. Actually, we publish too much, over 30 papers a year, but we try as often as we can to really publish in the good journals. And we have a number of good papers. They're always collaborative papers, many of them with the TwinsUK, where some kind of high quality biological data and samples is combined with our glycan data, and then we try to publish it as well as possible. And first direction where we went was, let's try to identify genes. Let's do the GWASs. They were extremely popular 15 years ago. So we published our first glycan GWAS in 2010, followed by the first GWAS of individual glycoprotein a few years later. And through a series of GWAS papers, I think we have over 20 of them now, we mapped a large network of genes which integrate to regulate IgG glycosylation. So there is no single gene for a single glycan, but this complex network of genes which decides about the glycan structure. And this is highly protein specific because we did parallel GWAS on 2 different proteins, transferrin and IgG, which have very similar glycan structures. So the structures are nearly the same. The enzymes which make them are nearly the same. But regulatory networks are completely different. So the GWAS hits which regulate glycosylation of IgG in a B cell and transferrin in liver cells are completely different. And we are now trying to expand this. For example, recently we introduced analysis of alpha-1 acid glycoprotein, which is the known acute phase protein, which associates with various inflammatory diseases with glycosylation changes, but it's extremely complicated and not much data. This is just first paper which we published earlier this year. So now we are trying to do the first GWAS. We have first 3,000 people analyzed, trying to generate a little bit more data to try to understand glycosylation of even more complex proteins. But GWAS is just the beginning because GWAS is generating hypotheses. For example, when we look at the glycan age clock of biological age, we have 16 genetic loci, which are the GWAS hits. But we then have to validate to see which of them are functionally relevant. And for this, we are using newly developed, we call them EpiCRISPR tools. So it's a CRISPR tool with deactivated Cas9. So instead of the nuclease, it has different effector functions, different domains, and 2 used here were VPR and CRAB. VPR is promoting expression of specific gene, CRAB is decreasing. So we developed a system where we can upregulate and downregulate a specific gene, and then in vitro look at the glycosylation, and we identify genes which in vitro have an effect of IgG glycosylation. And for example, one of the interesting stories is MANBA, which is mannose, which is not supposed to be working on IgG glycosylation, but it is a GWAS hit. So, and it is known to be a risk factor in different kidney diseases. And then we looked in vitro, okay, can we actually show effect of this gene in vitro? And indeed, if you upregulate MANBA, you also modify the IgG glycosylation. So it's an unknown mechanism. We still have to understand how it works. But this gene associates with changes in IgG glycan composition, associates with diseases related to changes in IgG glycosylation, and in vitro, it somehow changes IgG glycosylation. But there are so many potential questions because when we looked into pleiotropy, we have 94 different phenotypes which associate with the genes which regulate IgG glycosylation. So this will keep us busy for decades to come. Another large line of research which we do is more epidemiological, meaning looking into changes in IgG glycosylation and how do they correlate with other interesting things. And one of the first things we bumped into, and actually it was a problem for us at the beginning, because we were doing diseases and controls, and then we learned if we don't match cases and controls perfectly for age, we always see the same type of difference, and this is because IgG glycans change a lot with age. So this is 5 different cohorts, different part of the world, bluer man, redder woman, more or less the same trend in different glycans happens with age. And when we published that, the editors published editorials saying, are glycans the holy grail of biomarkers of aging? And I actually do believe they're right. And more we do, more we learn about the importance of glycans in aging. For example, when we compared 27 populations all around the world, we found that the changes in glycan strongly associate with the expected lifespan. So if you live in a country where you're expected to have a short lifespan, your glycans change much faster from young to old than if you live in the wealthier countries where your expected lifespan is longer. We also managed to generate some individual level data. This is still unpublished, but we have already 3 cohorts showing very clear association on an individual level of glycans which predict mortality and with not so small hazard ratios. So this is still unpublished, I cannot go into details, but what we have seen on the population cohorts, we already also see on an individual level where we have a long follow-up of people with samples collected before and actually waiting for them to die. And something we did together with Riccardo Marioni from Edinburgh on a Generation Scotland was to compare the prediction of mortality between the glycan age and just the classical glycan age, not optimized for mortality, and it's as good prediction of mortality as the GRIM age, the methylation marker optimized for mortality. So obviously, at the individual level, IgG glycan composition is relatively good predictor of mortality risk. And when we looked into the cases and control cohorts and we studied dozens of different diseases, in majority of them, we saw the change in the same direction as the effect of aging. So the last 4 bars are aging, and all others are different diseases from inflammatory, metabolic, HIV, and so on. And direction of change is more or less always the same. So the way the glycans change with aging, they change in the disease. So disease has some kind of an effect of accelerated aging. But what we actually think that we are seeing here is the functional role of IgG in regulating low-grade chronic inflammation, because low-grade chronic inflammation is an underlying factor in many different diseases, and IgG glycans are known to be regulators of low-grade chronic inflammation. And one of the strong indicators that this may be the case is that we see the changes in glycans up to a decade before diseases are actually diagnosed. So we did a number of prominent studies showing that glycans change in diabetes up to 10 years before there is even insulin resistance. Also, glycan seems to be the best predictor of cardiovascular events, both in primary and secondary prevention of diabetic complications. Something we did together with TwinsUK was the prediction of hypertension. Glycans change up to a decade before people develop RA, IBD, or similar disease. Glycans were predictive of severe COVID. And interestingly, glycans are one of the first molecules we change with onset of perimenopause. And based on all of this knowledge, we even launched the commercial test. So the glycan age is today available as a commercial test, which is monitoring anti-inflammatory and pro-inflammatory glycans, the way they change through age. At the moment, this is just a non-diagnostic test, but we hope to have it validated as diagnostic actually very soon. And of course, I believe you know that, but just to repeat, I am an inventor of the test. I'm a co-founder of a company. So of course, I'm heavily conflicted here. So you shouldn't just take my word, you should look into the data. But there's a lot of data. For example, this is a study done on, we did it together with Weill Cornell in Qatar. They wanted to measure more or less everything what can be measured. So they measured over 6,300 different traits, the proteomics, epigenomics, glycomics, and so on, many different things, metabolomics. And out of the 20 strongest associations with age, 9 out of 20 were immunoglobulin glycans, both IgG and IgA, while the others were mostly the sex hormones. And also, there is a lot of data showing that these glycans attached to IgG have multiple roles in regulating inflammation. And one of the best examples is IVIG therapy. So you get immunoglobulins generally from young, healthy people, put it in people with inflammatory diseases, and most of them will feel much better. So it's these glycans actually do have an anti-inflammatory effect when you take them from one person and give it into another. And kind of a big picture of this, this part of our work is that we are trying to redefine approach to medicine, because currently we go to hospital when something is broken. So when one of the organs stops functioning, we feel pain, and then we go to doctor and ask for help, and usually we get some kind of a pill. And these pills generally do not cure the disease, they just alleviate the symptoms. So we become the lifelong customers of a big pharma. And this is an endpoint. This is beyond the point of no return. But this takes up over a decade to lose the homeostasis, to do all these molecular changes which actually lead to breaking down of some of the systems. And if we can monitor these changes early, and immunoglobulin glycan seems to be a very early indicator, if we see this change on time, then maybe a simple preventive intervention, like a weight loss or moderate exercise or early pharmacological therapy, can stop this trajectory and return people to homeostatic conditions and either completely prevent or at least postpone disease development for a number of years. And we have several nice examples. One of them is cardiovascular diseases. So the initial study was done together with the TwinsUK and Edinburgh cohorts, the Jim Wilson, where we saw the strong associations between IgG glycom and cardiovascular disease risk score. Of course, this is just an association, but we used it to get the samples from the EPIC cohort. EPIC was collected over 30 years ago, and we got samples from the German subsection, 27,000 individuals. Out of them, approximately 500 had either heart attack or stroke. And when we compared them to the matching controls, in men, IgG glycom was as predictive as the entire AHA score. In women, it was even more predictive. Actually, even the single glycan in women was more predictive than the AHA score, and with a hazard ratio, which is not that small, 0.7. Of course, this is one study, even though it was published in a good journal, we wanted to replicate. So we did another cohort, again, got a very similar hazard ratio in women. Interestingly, again, completely different glycans in men, a little bit weaker prediction, but still reasonably good. And just recently, we published the second replication with 2 additional cohorts together with a colleagues from Harvard, which was published in Circulation Research. Again, the hazard ratio around 0.7. So more or less the same thing in the 4 cohorts so far. So we believe really IgG glycom is predictive of CVD events. And maybe there's a way to hack it, at least in mice. So if you feed mice with a MANAC, which is the precursor of cellulation, you can prevent hypertension in obese mice. So you put mice on high-fat diet, they will become obese and they will develop hypertension. But if you give them MANAC as a supplement, of course, they will become obese, but they will not develop hypertension because their IgG will not lose their cellulation, which has an anti-inflammatory effect and somehow affects the endothelial biology to prevent obese mice to develop hypertension. Another interesting line of research, again, coming from collaboration with the TwinsUK, is the prediction of diabetes. So here, it is not the IgG glycom, it's the total plasma glycom. So the glycans coming from all plasma proteins, just cut them off and analyze them. And when we look into individual glycans, so this is the insulin resistance, this is the healthy state, 10 years ago, 7 years ago, 4 years ago, insulin resistance. So we can see glycans changing in a gradual way from kind of a healthy state to development of insulin resistance. And we know that these glycans are can be found on the different receptors, including the glucose transporters, the insulin receptors, and some other membrane proteins. So, and this is why it's very interesting to see that exactly the same glycans which predict development, which kind of track development of insulin resistance, are also predicting the postprandial response in both lipids and glucose, although glucose, of course, extremely difficult to measure. Again, this is a study we did together with the TwinsUK. And the most interesting thing of all is that if you change your lifestyle, if you lose weight, these glycans, which track the progression towards insulin resistance and diabetes, will go in the opposite directions. They will kind of have a decrease in a disease risk. When we compared these changes in accelerated aging in glycans with different other epigenetic clocks, interestingly, they do not correlate. So there's very little correlation in acceleration of glycan and epigenetic aging. This was published by the Edinburgh team 2 years ago, and we recently did a replication. This part is unpublished, but when you look at the clock itself, of course, they correlate because they all correlate with age. But if you look at acceleration of a glycan age and all other epigenetic clocks, there's nearly no correlation. These are different aging mechanisms. And we, of course, we still don't understand these mechanisms because we don't understand aging in general. But some interesting insight came from our study of the large cohort of people with Down syndrome. Down syndrome is known to be associated with accelerated aging. And if you take a cross-sectional cohort, you will see that people with Down syndrome look older than the people, than controls. But this acceleration is actually happening very early in life. Because when we look at the trajectory of aging in controls and Down syndrome individuals, we see that actually the pace is the same. Just there is an acceleration early in life. And it's a very complicated paper. We published it last year. So if you want to go into details, this is the paper. But through some individuals with only partial trisomy, we managed to pinpoint this to DYRK1A kinase, which is linked to the unrepaired DNA damage, which in early life seems to be more detrimental, of course, than later because there is more cell divisions. And this DNA damage may be the driver of glycan aging, at least one of the drivers. And additional evidence in that direction is when we looked into the people on antiretroviral therapy, so HIV positive individuals who were fully controlled with drugs, so there is no active virus, but we see a rapid acceleration of glycan aging, even people with a full control of the virus, which may be related to the side effect of antiretroviral drugs, which sometimes do cause the DNA damage. And I think the most important question would be, are these changes in glycans something that we can revert? Can we revert the glycan age clock? Can we slow down this inflammation and postpone diseases? And we did a number of intervention studies looking into the physical activity, dietary interventions, sex hormones, some drugs, and indeed, glycans are modifiable, much more than any other biomarkers of aging which are currently being used. And approximately 40% is heritable, 60% is something which we can actually act upon. And of course, there are drugs which work. So if you put somebody on anti-TNF biologicals, there will be a significant reduction in inflammatory potential of the IgG glycom. But of course, this is something what happens in inflammatory diseases. You, of course, don't give anti-inflammatory biologicals as prevention. But some people do strange things. And we were looking, for example, in a plasma exchange. So this is one of the known rejuvenation therapies. You take plasma from young individuals, put it in older individuals. So this was a small group of humans who do it. And we did see, of course, an improvement in the glycan age. And of course, it's extremely preliminary, but this was a study published actually this year in Geroscience, where we joined kind of at the final moment. This was Steve Forward who organized it. So they were separating plasma into fractions, and one of the exosome fractions from newborn piglets was given to the rats, and the rats had a rapidly decreased epigenetic age. We looked into glycan age, and glycan age also rapidly decreased. So these were rats 6 months apart. This is the regular aging of a rat, and this is the rat put on this exosome fraction, which actually went backwards and not regular aging. So there was nearly a 1 year effect in 6 months. And these rats actually live longer. So there is something in the exosome fraction which affects aging and which affects IgG glycosylation. But as I said, one study so far, rats, it has to be studied further. Something we know has a very strong effect on the IgG glycosylation in human is estrogen. Estrogen, and in both men and women, is one of the key effectors. This was a very cool study done nearly 20 years ago in US. So they had approximately 50 women with a chemical-induced menopause, so no sex hormone production. Part of them had the estrogen add back, the other ones get placebo, and the placebo group aged on average 9 years within the 6 months. After the end of the therapy, they return backwards. There was a parallel group also with men, and in men, testosterone, of course, works, but only works if it's being converted to estrogen. So if you block aromatase, you block the conversion of testosterone to estrogen, you don't have this beneficial effect in men. When we tracked the woman through menopause, and this was again done together with TwinsUK, we realized that there is really a rapid acceleration of glycan aging at the time of perimenopause. And actually, this is one of the first symptoms which we see that somebody is approaching perimenopause. And we believe that this is the case because actually IgG integrates estrogen concentration of a couple of weeks, because estrogen is extremely difficult to actually, it's not predictive because it can change dramatically within a day and between days. You have to measure it in multiple days to know the average estrogen concentration in an individual, and IgG is doing it for us. So it's kind of an analog of HbA1c for glucose. And this is something we are now trying to position as a diagnostic marker. And we also did a very complicated study where we had the 50 women giving us weekly samples for 12 weeks. And you can actually even see this variation through the cell cycle, through the menstrual cycle, but the variation is relatively small. So it's usually approximately 1%. So it's not a problem in a cohort studies. We are also testing a number of different so-called aging interventions. Some people take metformin preventively. And we looked into one of the cohorts, and only one individual actually did have effect. Majority of them didn't have effect of metformin. So our conclusion here is that metformin is not an anti-aging drug. It's not helping healthy individuals. But if you're a diabetic, then of course metformin will help in a much larger proportion of cases. So here we compared metformin, SGLT inhibitors, and GLP-1 are agonists, and each of them will have beneficial effect in some individuals, but not really beneficial in others. So things has to be personalized. Weight loss is working. So bariatric surgery, aggressive but effective. We had people improving for over 30 years in 6 months. Majority of people improved, not everybody, but again, did not want to go to surgery if it's not necessary. So we checked other interventions. Caloric restrictions seem to be working. So here we had 1,000 people on 800 calories a day for 2 months. So it worked, but that's torture. No, nobody can stay on 800 calories for longer. Although there was one study in US where they had people on a low-calorie diet for 2 years, and we do see effects. So this is a small pilot, which we did. So this chronological age, this is what happened with the glycan age in these individuals. So we do see an effect of prolonged calorie restriction in reduction of glycan age. But as I said, calorie restriction is not ideal. So let's try to find a diet which works. And through the Diogenes study, we tested 5 different diets, and each of the diets, some people would get better, some people would get worse. So there is no magic diet. There is no one-size-fits-all approach. And I think this aligns very well to other studies that you have done. And we really have to find a personalized way to diet which would work. One good news from studies we again did together with TwinsUK is that whenever you start losing weight in the 30s and the 40s and the 50s, you will decrease your pace of aging. So it's never too late to start losing weight. We've been made believe that you can sweat out the bad diet, but this is very wrong. So physical activity is of course necessary, but you cannot sweat out the bad diet, and physical activity is actually more often pro-inflammatory than not. We did a number of studies. This is one of the studies where we managed to get an anti-inflammatory effect of exercise, and this was high-intensity interval training. But when we did a huge study. So we took a couple hundred people into exercise training, middle-aged, obese individuals in Zagreb. So we put them in the exercise program. And initial 3 months, they all got worse. And after 3 months, we said, okay, maybe they're not exercising enough. Let's do it longer. Let's do it harder. So they did an additional 3 months and then additional 3 months. And even after 9 months, on a population level, we saw very little beneficial effect in the glycan age. So it's most of them were actually overtrained. So what we learned from here is you cannot just take a middle-aged couch potato and force them to train. So one good thing which came from that study is there is this glycan which predicts heart attack and stroke, and it did improve. So this is the protective. So it decreased the risk of heart attack and stroke in all types of exercise that we did. So exercise is good, but it has to be carefully optimized. And this is an interesting study we recently published just earlier this year. So this is the average Croatian people, which is obese and inactive. And when we try to push them to exercise for 6 months, actually they got worse. But when we looked at the matching group of regularly active individuals, they turned out much younger. So regular physical activity is beneficial, but too much is detrimental because these are professional athletes, actually even a bit younger than this group here. And they got even worse than they looked worse than the inactive population. So physical activity is good, too much physical activity is not good. But the problem is, who can really tell me what is the best for me? Average person does not exist. Everything we know is based on an average person. And this is where we are trying to position glycan biomarkers. And I think we are doing it pretty well. There was a recent Cell Perspective paper by the Biomarkers of Aging Consortium, and they clearly said that the glycan age is the only commercially available test used to track response to lifestyle changes, and they do associate with the multiple diseases. Kind of a big idea is let's try to catch people early before they go into disease. We know that we have to do preventive maintenance of our cars. If we would wait for a tire to explode on a highway, driving would be very deadly, but we do it to our bodies. We wait for something to explode before we try to fix it. And again, this was generously funded by a number of grants, which I have to acknowledge. And I have a large team who is actually doing all the work, so I can not travel this time, but at least talk to different people. And I thank you all for your attention and more than happy to answer questions.